everyone, and you are very welcome to this TU Eindhoven webinar on how to become a tissue engineer. My name is Dr. Barry Fitzgerald, and I'm a science communicator here at TU Eindhoven. And today, I'm very happy to be joined by two tissue engineers who are based here at TU Eindhoven. First of all, I have Associate Professor Sandra Hofmann. You're very welcome.
Welcome.
And Assistant Professor Anthal Smits, also in tissue engineering. You're very welcome, Anthal.
Thank you.
So tissue engineering is a fantastic career. I have to say I'm supremely jealous that the both of you are working in the area of tissue engineering. And I just want to refer back to something that actually Sandra pointed out to me over the last couple of weeks, that back in 2000, Time magazine published an article with the hottest jobs of the future. Now, the top three I thought were very interesting. There was no sign of machine learning, algorithm programmers, nobody working on AI. The top three were farmers, spelt with PH, so obviously something related to pharmacy. The second one was gene programmers, which given the rise of CRISPR-Cas and other genetic engineering tools, isn't a bad prediction. Number one is tissue engineers.
Yeah.
So congratulations on
picking
the number one on the Time magazine 2000 hottest jobs list. Now, many researchers nowadays are working in the field of tissue engineering, and we're going to hear a little bit about what both of you are doing in terms of your research later on in this lovely discussion we're going to have. But first of all, let's get to know Sandra and let's get to know Anthal and find out a little bit more about how you got to where you are. So this is all about your career, and these questions I'm going to talk about basically how you got to where you are right now. What was the trajectory that you took? What were the things that you encountered? What were the lessons that you learned? So why don't I start first of all with Sandra. So why don't we get you to briefly say a little bit about yourself and where you're from?
My name is Sandra, obviously. I'm Swiss, that's important to know, I think. I grew up somewhere in the Alps, and I studied something that people would probably not associate first with tissue engineering. I studied pharmaceutical sciences. So I've always been interested in health-related issues, but I knew I didn't want to become a doctor, and chemistry and biology were kind of too far off. So that's how I studied. And I did my master's thesis on a project that was related to bone tissue engineering, and it caught me. And actually, towards the end, I was still doubting whether or not I wanted to do a PhD or not. But I got the opportunity by my former supervisor, my master thesis supervisor, to join him to Boston for his postdoc, because he said he could use a helping hand, and why I wouldn't join him. And I was finished with my studies and thought, yeah, why not? And he said, it's going to be very fancy. You're going to be working with bioreactors. And I thought, I have no clue what this is, but it sounds good.
So just so people maybe explain, what is a bioreactor?
Yeah, well, it can be very simple. It can just be a dish where you can cultivate cells in it, but it can be very fancy, that you can do advanced imaging with it, you can mechanically load your samples, but it can also just be a petri dish, basically.
So you went to the US and you worked on bioreactors in Boston, and then what happened next?
So I came back, and basically I started my PhD at ETH Zurich. It was on silk fibrin as a scaffold for tissue engineering and for drug delivery. So it was still somewhat pharmaceutical science related.
So was that spider silk or was it silkworm silk?
Silkworm silk.
Silkworm silk.
Yeah.
I personally would love it to have been spider silk, but...
Yeah, well, you could probably do it, but it's a bit harder to get in reasonable amounts to produce.
Absolutely, yes.
To produce large scaffolds. So that's what my PhD was about. And then there was this decision about postdoc, yes or not, and I had actually accepted a postdoc back in the US, but then I also had, my father had a heart attack, and it didn't feel correct to move so far away from my family. And I had another offer back in Switzerland, so I decided to stay in Switzerland, did my postdoc there, also in tissue engineering actually, but then with a different focus, more on bioreactors again, and also on advanced imaging. And and I stayed within that group, or was allowed to build my own group actually within under that professor.
Mhm.
And in 2013, I made the step from a group leader to an assistant professor here in Eindhoven.
Okay.
Yeah.
So you've been in Eindhoven since 2013 then?
Yes.
So coming from Switzerland and you moved to the United States for a period and then back to Switzerland and then to the Netherlands. So you've traveled a little bit. What for you are the big differences between, let's compare Switzerland and the Netherlands? Now I can think of one very obvious one.
It's a bit flatter here, yes,
Yes,
obviously.
there we go, right?
Yeah.
But in terms of the work ethic and the groups and how people interact with each other, have you noticed any, for you, cultural differences or changes?
I would say we're pretty similar, actually. I think I can understand well how hard it is to make friends, to get friends, because people are very close with their friends from very early on. Like many have their friends from school times, so if you come in later, it's a harder time to make friends. But I would say it's similar in Switzerland. I must say, coming here with kids, or I had one kid when we came here, and my second son was born once we were here. I think combining work and private life is easier.
Okay.
Yeah.
So then moving on to someone who is from the Netherlands, Anthal, can you just tell us a little bit about where you're from and what did you study at university?
Yeah, so I'm Dutch.
What part of the Netherlands are you
I come from the south, from Limburg,
Okay.
and I've lived in Eindhoven for about 20 years now. So I'm a very much homegrown product, you could say. I studied here in university, but I studied mechanics first, mechanical engineering. And during my bachelor studies, I always had this fascination for the human body and how it works. And I felt that during my bachelor's in mechanics, it was fun, but I missed this part. And I started taking up some courses in biomedical, extracurricular, and taking up some projects. And then this was pre-bachelor master structure. But then I decided to switch to biomedical engineering. So I did a master in biomedical engineering. I did hardcore biology projects to compensate for my engineering background on cardiomyocyte progenitor cells and regeneration started back then. And after that, I rolled into a PhD.
I like the phrase rolled into a PhD.
This is how it happened.
Is that how you would describe deciding to do a PhD? I just happened to roll into it.
No, I was at that point. I knew I was pretty sure that I want to be in science, at least at that point. So that was a natural step, and there was a super cool project that, yeah, the opportunity was there, so I jumped on it essentially. And then during my PhD, I started working on really tissue engineering and in situ tissue engineering, and I developed a fascination for the immune system and how this works. And I concluded my PhD in 2014, and then I worked for a company for a while, for LifeTech Group, which is an R&D company that does contract research for mainly for people that develop, for example, biomedical devices or therapeutic strategies. And they work a lot with ex vivo platforms, so culturing tissues in the lab and then try to do a certain therapy on there.
And what was the biggest difference you found from moving from the academic world to the industrial world?
Yeah, that was quite a big step for me because in industry, of course, it's a profit organization. So I did a lot of different projects at the same time, short term, which is nice, right? You get to work on a lot of different applications. If one project has your interest a bit less than another, it doesn't matter so much because you'll switch all the time. But I missed the depth, right? So really following your curiosity and just following your nose, basically, what you do as a researcher.
Yes.
This is something I missed in the company. Although it was very innovative and fun to work there, it's a very small company. The atmosphere was great, but like my heart lies in research.
In research in terms of longer than a 6-month project?
Yes, for sure.
Like a 10-year project or a 20-year project?
Yeah, and not necessarily in time, but more also in depth, so that you wonder about things and you can figure out how stuff works, basically.
Yep.
So after having worked 2 years for the company, some of the work of my PhD project here at the university that got funded by some bigger consortium projects, and I sort of sensed that somebody with my expertise on immunology was missing, or I should say the combination between immunology and engineering. And that's why I took the chance basically to quit my job at LifeTech and then jump back into academia.
And what was that transition like, moving from the industrial world back into the academic world?
Yeah, it was easy in a way because I was already familiar here,
Sure.
of course. I came back with a very clear goal in mind, so not just to do a postdoc, but really to get tenured. And of course, there was no tenure track at that moment.
Sure.
So it was a bit of a gamble, let's say. But I immediately got to work with some very nice PhD students that were working on these projects, and I knew them a little bit, so we stayed in touch. So this was very refreshing in a way, and I think I got back on track quite fast. Yeah.
That's good. A question that's motivated by Anthal's experience is that, did you ever think about, Sandra, looking at the industrial option at any point in your career?
Well, with pharmaceutical sciences, I could work in a pharmacy. I worked for half a year in a pharmacy after my PhD, just because I had to bridge some time.
Yep.
It was never as much that I would have jumped and say, yes, I want to try it. But I'm always thinking, maybe I would like it if I did it. But I cannot say because I don't know.
Exactly, of course. You make a decision and that's the path you take.
Yeah, but I'm too much loving what I'm doing right now to
try it.
To try it, yeah.
Okay.
Yeah.
I've got an audience question here in regards to the content we've been talking about, and that is, for both of you, maybe you face the same obstacles in getting to where you are in your career. What would be, for either of you, the biggest obstacle that you encountered as you moved through this progression in both your careers individually? Maybe you have the same obstacle, I don't know.
Yeah, well, there are a few things that are challenging in general when you're in academia,
Yes.
like getting funding and so, and I think everyone knows that, everyone does that.
I think there are many researchers around the world who could appreciate that comment, yes.
Yeah, I think for me, the bigger obstacles were more like, the decisions that you have to make together with your families, like me not going for a postdoc because of health issues within the family, or moving with a whole family to a foreign country. I think that were more obstacles, and if your partner doesn't support you in that, then that's basically it. Yeah.
I totally recognize that.
Yeah.
So for me, one of the big obstacles was because I came from industry. It was only 2 years, so it sounds short.
Yes.
But it sort of disqualifies you for certain grants, early stage scientific grants. And because basically normally the normal trajectory would be to go after your PhD, go abroad, do a postdoc, and then come back and get some experience in a different lab. And of course I didn't have this. And at that time also my wife, and I have two small children. So that was a tough decision. I juggled with it for a while, to move, for example, to the US for a while, or not. And I basically replaced it by doing shorter trips back and forth, which is very tiring, but you can at least...
you still get some experience of doing and working in an international setting.
Yeah, and I think it's important to mention that there's not one track, right, to get where you are.
Absolutely.
So even if you have these struggles, you can find another way that would work.
Because you have a background in mechanical engineering, and Sandra, you have a background in pharmaceuticals, but you're both tissue engineers.
Yes.
So neither of you, the dream at the start was not to be a tissue engineer, necessarily so.
Oh, at the start of my studies, I had no clue what tissue engineering was. I think, that was in 1997. That was when tissue engineering was born. So,
and that's why it was highlighted, let's say, in the Time magazine in 2000, because it would have been one of the hot topics, emerging topics at the time. Right, so why don't we get into a little bit of your research just to get a flavour for the type of things that you're working on here at TU Eindhoven. But at the start, I just want to do like a very quick fire thing on some technical terms. You've both mentioned a couple of them. So I'm going to give you like a couple of moments very briefly. We'll alternate between the both of you to explain some of the terms. So first of all, what is ex vivo?
Ex vivo is outside of the living. So for example, if you take a piece of bone out of the body and you look at it outside of the body, and ideally you keep it alive for a while.
Mm-hmm.
Yeah.
And that's very much in line with some of the things that you do.
Yes.
And then there's in vitro. What does in vitro mean, Anthal?
In vitro means in the laboratory, so that's very straightforward. If you culture cells or tissues, we call it in vitro.
And then you have ex situ or in situ. How would you describe both of those terms?
Do you want?
In situ is in the functional site of a certain tissue. So.
So in general terms, it basically means that.
Yeah. So for a heart valve, it's where the heart valve is in your body. So in its site, in its own functional
So
site.
it's basically if you were putting a heart valve in the body, it's to test it in its natural environment where it actually should be in the body.
Yeah. So you can put tissues or materials under the skin, which would not be in situ.
Okay.
But you could also put it in the site where it actually should be.
And then in vivo.
In situ.
And the difference between in vivo, maybe some people might think in vivo sounds maybe a little bit like in situ, but in vivo is.
Yeah, it is inside the living. How to distinguish it from in situ is basically if you, an in vivo study is following how patients are doing, for example, whereas it doesn't have necessarily to do with anything implant.
Okay.
Like.
Okay. These are just some of the terms that sometimes people who are interested in tissue engineering or in those type of areas come across, and it's great because at the start you mentioned each mentioned one of those terms. So what we're going to start with first of all is Sandra is going to describe some of her research, and if you want to take over, you can just describe some of your research.
Yeah. So I'm pretty much interested into bone. How does bone work? Can we understand how bone works? How does it regenerate after a fracture, so after it is broken? And also, what goes wrong when bone diseases develop? And for that, what I'm interested in is actually the main contributors of bone, or in particular bone remodeling. And in my view, these are the bone cells. These are the active parts. So these cells, they are actually capable of either putting down bone where it's needed, so produce new bone where new bone is needed, or taking away bone where it's no longer needed. And this process we call bone remodeling. And we have three main contributing cells. So these are, on one hand, osteoblasts, the cells that form bones. They actually, once they embed themselves in their own bone matrix, we start calling them osteocytes. And the osteocytes, they are embedded in their matrix, and they are more or less the regulators of the two other cells. And they can actually also recruit the osteoclasts, so the third player, which are the cells that are capable of removing bone. And it's thought that the osteocytes, they act and react when they are mechanically loaded. So if you have a bone that is no longer loaded, like bones of astronauts, then actually the osteocytes can sense that, and then it can signal to the other cells, hey, we have a bone here that is no longer needed, come take it away. And then they recruit or ask the osteoclasts to act. And actually these three cell types have to be in a very nice balance so that our bones are in a homeostatic state. So, and in many of the bone diseases that we know, they are out of balance. So osteoporosis, for example, the osteoclasts are too active in comparison to the osteoblasts. So there is too few bone mass left because they are too active in taking away bone.
Okay.
So that's what I'm pretty much interested in. And if we try to translate that to the tissue engineering context, what I try to do with my group is actually we try to grow little bones in the laboratory. And we actually do that with the typical components that people use in tissue engineering. It's a scaffold, a three-dimensional structure where cells can attach to. So a scaffold provides an outer shape, a surface where cells can attach to. And then we add cells and give these cells certain signals, and so that these cells actually start becoming what they should be. We usually start with progenitor cells, or stem cells that can become osteoblasts or osteoclasts or osteocytes. And we give them chemical signals, can be growth factors. We give them mechanical signals, so we stimulate them with, for example, bioreactors. And we follow over time how they're actually growing, depositing a tissue, or in the case of osteoclasts, how they are removing tissue. And what we would like to achieve is a tissue-engineered piece of bone that does not only look like bone, but actually acts like bone, functions like bone. So if we started, for example, with a deceased cells from a patient, we would expect them to see also the disease in the tissue-engineered piece of bone that we build.
Okay, so that's a fantastic introduction to your work. There's lots of questions I could ask. I'm going to ask two very quick ones, and we'll get on to other ones later on. The first one is, so you're trying to grow bone in the laboratory.
Yep.
And I'm really interested in this because I asked you this before, and I'd like to ask the question, is that you're working on bone, but were you motivated by breaking a bone in the past?
No.
No.
No, I don't know. No, I never broke a bone myself, neither did someone close to my family.
Or do you know anyone who has osteoporosis?
Yeah, also not in my family,
Okay.
no.
So
no.
what was the closest you came to seeing a broken bone? Or
Yeah, once I played football with a friend of mine, and she broke her bone so she was the goalie, and I probably was hitting a bit too hard, or she fell, and she broke her arm.
It's a contact sport, so it's,
yeah.
you know.
Yeah, so that was the closest to bone, but actually that was a simple fracture.
Yes.
What is fascinating though is that bone has a high regenerative potential. So if you just put the two bone ends together, it can heal perfectly.
In what type of a time period?
That depends a lot on the fracture and,
Yes.
and on the bone, but within a few weeks you're ready to go.
Which is quite
again.
extraordinary.
Yeah.
So that leads on to my second question. What for you is the most fascinating thing that you've learned about bone in your work?
Yeah, actually it is, and how the cells interact with each other. And they are, yeah, you can look at them independently, but more I come to believe that, yeah, we know it's their balance, but we really need to look at how they interact with each other.
Okay.
And I'd really like to understand how they talk to each other and how they motivate each other to do what they're doing.
Okay,
Yeah.
very, very good. Anthal, I'm going to ask you to introduce your research very, very briefly.
All right, so what we focus on in my group is mainly cardiovascular regeneration. So our main applications are heart valve prosthesis and small diameter arteries. So, and this is driven by the clinical need for these devices, basically. So a lot of people get a heart valve transplant or a bypass surgery or a stent.
Mm-hmm.
And some of these devices in some applications don't work that well. So we try to improve them. And the technology that we use is, we call it in situ cardiovascular tissue engineering. And this is basically, we implant a scaffold without any cells on there, typically, directly. And we try to trick the body into thinking that it should replace, on this side, a new heart valve, for example, or blood vessel. So we make scaffolds. They can be synthetic polymers or natural polymers that are degradable, like your sugar cube in your coffee. It's there only for a short time. And then once we implant it, it should ignite an inflammation, which is the trigger for your body to make new tissue. And what we mainly study is how to harness this inflammation. So you can compare it a bit with if you have a splinter in your finger. It always turns a bit red first before it heals. So this is basically the inspiration that we need this to trigger this inflammation, and that will trigger
So in many ways what you want to do is you want to trick the body into thinking that it is actually damaged.
Yes.
To initiate and kickstart the healing process, the first stage of the healing process, which is inflammation.
Exactly.
Many people might think that that's contradictory. Why would you want to hurt the body at first, but as you've
Yeah.
said,
Well, this is a good point
Yeah.
because at first, the first prostheses that were made were always, people tried to make them inert, so as invisible as possible for the body. But this is simply impossible. Your body will always detect that something foreign is there, that is not supposed to be there. We try to make use of the capacity of your body to regenerate. And for heart valves, for example, this is very poor, unlike bone. But if you give the right cues, if you trick the body to think that it can regenerate or that it needs to regenerate there, then it's a very powerful tool. And then, so instead of course, we don't just put tissues in and we watch what happens, but we really try to understand what happens in the body to a material that we put in, how the cells respond to it, how they break down this artificial material, and how they build up new tissue. And for that, we make use of bioreactors, they were mentioned before, in the lab, where we, for example, use our scaffolds, our heart valves, or little pieces of scaffold in models where we seed cells on there, either from cell lines or patient cells, and we see how they respond to this material. And we hope that by that we can predict what will happen once we implant the material.
Okay, excellent. So I'm going to ask a couple of questions just based on kind of the crossover between your work. You both mentioned scaffolds. What type of scaffolds do you use in your work, and can you just describe the importance of scaffolds in tissue engineering? Maybe Sandra first.
Yeah, so I'm mostly using silk fibrine scaffolds, the ones that I mentioned before. Not because I particularly think they're the best for regeneration, but they do a good job for the moment for what we're doing. We're also looking in alternatives like taking pieces of trabecular bone and using these as a scaffold, because this would be a more natural environment for the cells. So one is the very natural environment and the other one is well, the silk fibrine, it's a protein. The main protein in bone, or one of the main extracellular matrix components in bone, is collagen, and silk and collagen actually have quite many things in common. So it's not that far off using silk, I would say. We mainly need it to be very porous, highly porous structures that can give an outer shape, like it can give the shape of the scaffold, so that the cells do not just clump and sit as a very small ball somewhere in the edge. Yeah.
Okay, and Anthal, you use scaffolds as well. What type of scaffolds are you using, and how important are they for your work?
Yeah, very important, clearly. So for us, the scaffolds have to be functional, because in the initial stages there are no cells, there's no tissue. So if you implant it as a heart valve, it should function as a heart valve. And that means that its mechanical properties are very important. So it shouldn't break, for example, simply said. We use mainly elastomeric plastics that are degradable. And we are fortunate enough to work in the ICMS, the Institute for Complex Molecular Systems. And we collaborate with Professor Bert Meijer and Professor Patricia Dankers. And they have developed these supermolecular materials, which are sort of like, it's sort of a Lego kit with different building blocks that you can combine. And with that you can play with how fast does it degrade, how strong is it, can you put bioactive parts in it, all that kind of stuff. So it's, we have a sort of toolbox of ingredients that we can use.
I assume depending on where you put scaffolds in the body, you want the scaffolds to be there for a certain period of time. So in certain parts you want it to be there for a short time, maybe for other parts a long time. So are there polymers like that that can change the amount of time they need to be in the body?
Oh, yeah, this is a very interesting question. For sure this is possible. To match, the biggest challenge for us is actually to match the degradation of the scaffold with the tissue that is formed. So if it degrades too fast, but there's not enough tissue yet, your organ will fail. And if the scaffold stays there for too long, you get chronic inflammation, you get fibrosis, you get a scar, essentially. So to match this is very difficult, especially because it can vary from patient to patient, how fast the tissue will be built. So what we mainly study is this balance between, and so cells are needed to degrade our materials. They don't just disintegrate over time, necessarily. And we need this to be an active process. and cells also regulate the tissue formation. So this is how we try to balance this.
So I must say that for both of you, you're both trying to grow tissue in the laboratory. How do you remain so patient with this type of work? Because I have a computational physics background, and when I would be writing computer programs and they don't work, and I can just stop them, change a variable, and then restart the program. But in your work, it's not like that, right? Because growing cells in the laboratory can be a very testing, I can imagine, period of your research.
Yeah, well, but you can also say, I think that's a bit different to the computer world than is with every failed experiment, well, unless you have an infection or so, I think you can also learn.
Absolutely,
you can learn
yeah.
a lot, and you can learn what to do, what not to do. It's indeed sometimes hard to see it in that way, like a learning lesson and something positive, but I think if our goal is to ultimately understand what the cells need in order to perform in a certain way, then you can actually learn a lot from failed, so-called failed experiments too. It's just not as easily publishable, which is then very frustrating, especially for those that need publications to finish their PhD, for example.
I can understand that.
Yeah.
Of course, I would have encountered failure as well, and I would know this variable doesn't work, but the lesson I learned is over maybe 1 minute, while in your case it could be weeks of work that has gone into culturing cells. Is that correct?
Yeah, well, our experiments usually take along 4 to 6 weeks.
There you go,
usually,
yeah. And
Yeah.
Anthal, with heart valves?
It depends on the experiment, but they are a bit
Yeah.
shorter, but
Yeah.
in the same
Yeah.
line.
Yeah.
You need some persistence, I think.
Yeah.
That's for sure.
Do you also echo Sandra's thoughts that failure is a good thing?
Definitely, yeah. Actually, yeah. So, in the in vitro experiments, well, I had a PhD student before that, and came into my office. I was very depressed because the results were not as expected, and then I always got super excited because then, if everything was as expected, then we wouldn't have to do the experiments. So I think this usually fuels new research, and typically the most interesting things are serendipity findings that you didn't foresee. So,
absolutely. And we've got one question coming in from the audience, and somebody wants to know what's the most complex part of making heart valves.
Ooh. Well, the most complex part of making heart valves is that heart valves have a very particular structure. So they're not just flapping around in the blood, but they have 3 layers, and within these layers they have a very specific organization of the proteins that are in there. For example, collagen, but also elastin. And this gives them a certain mechanical properties, but especially also durability. So this means that your valve can work as long as you live, basically, and that it can adapt if your blood pressure goes up slowly when you age, which is also why I'm so fascinated by this tissue. But this is also what makes it very tricky to regenerate. So we can easily make a collagen heart valve, just one protein.
Sure.
But to get the right organization in every layer, that is very difficult.
So then the answer is there's lots of things that are...
For sure,
yeah. So I just want to move on to the fact that you both have research groups here at TU Eindhoven, and you have PhD students, and you have a number of projects running. I think it'd be nice just to think about what's it like to be running a group and having research projects, and in terms of the work that you're doing, do you always have a set goal, or does that goal change? And is it malleable, and can it be you're not really fixed on something during your research process?
Well, the overall goal is more or less given, but the way to get there is for sure changing. And in terms of having a research group, I think I saw my own change quite a bit, because for others that might be different, but I was not like, I want to become a professor and that's my goal. It was more like I was into research and I liked that a lot. And then I kind of got into having students and teach them something. So I also started giving what I knew further to someone else. And somehow when you go up the hierarchy, so to speak, then at some point you actually find yourself not doing any work in the lab anymore. At least if I'm in the lab, I'm usually showing around visitors. So I'm not doing the work myself anymore, which was the reason why I initially started, because I like to do that. And I think with that also, my whole position changed. And it took me a while to make this switch from, oh, another student walks into my office and wants something from me, to, so now my job is actually to raise these students, to fascinate and motivate them for that research that I'm burning for, but they should be doing it for me and for themselves. Yeah.
Do you miss the laboratory?
Yes, I do.
Is the same apply to you, Anthal?
Yes.
Do you miss the laboratory as well?
Definitely.
Yeah? So you do not, both of you don't spend as much time as you did in the past in the laboratory because of the tasks and the commitments to running so many different research projects or PhD students.
No, your responsibility shift.
Yes,
Yeah.
yes.
Very, very straightforward.
And when you find something interesting, if you find something interesting that you didn't plan or envisage that you'd find, do you allow yourself that little bit of flexibility to be able to go and explore it? What if it was something astounding and you think, wow, but this might take us 4 months, 6 months, 9 months off the project trajectory? Do you allow yourself that, or do you have like criteria to think about, is this really worth exploring?
I definitely go along the sides. We often do that with student projects.
Yep.
Because if it's too risky to put a PhD student on that has to finish within a certain time, it's usually okay to have a master student explore it under the guidance of a PhD student. So at least the way I do is I usually try to persuade one of my PhD students to also believe that this would be very interesting to go for. And that works well.
That's good. That's good. I've got an audience question, and they've asked, how close do you think we are to engineering soft tissues, full soft tissues now, like hearts, lungs, kidney, liver? Just both of you being tissue engineers, let's think about this in terms of addressing organ requirements and organ transplantation
these
requirements.
are organs. Yeah.
Yes, they're organs, yeah. What do you think? Are we days away or are we decades away?
Well, I think, but more in general, the field of tissue engineering has changed a bit to think more, for example, this Time magazine article you mentioned. At that time, tissue engineering was more of a hype, right? So people were showing, look, we can make lungs, we can make, or whatever, kidney in a small dish. And it was for showing off, basically, because you can do it. And now I think research is shifting more to asking different questions, like understanding processes, as Sandra also mentioned, really for translation, so really to make it a therapy. And that has changed, for example, for us, that we are not culturing the perfect heart valve in the lab anymore, but we are trying to seduce the body to do it. But you could apply this like paradigm shift to different tissues. I think we are getting there, right? We are building understanding. It stays very difficult to control cells in the lab because, I mean, the more you know, the less you know. The more you know, you don't know anything. So really to get this hierarchy and structures in tissues, that is still very difficult, I think. But yeah, well, we'll get there. Yeah.
Sandra?
Yeah, I fully agree. That we already fear that we agree too much on things, but yes, I fully agree.
Yeah. Well, here's a question where you might have a different
Disagree.
opinion. Maybe you might disagree. I don't know. So I'm a big fan of superheroes and superhero films. I watch them a lot. There are many characters who have the ability to regenerate over timescales that are, well, let's say a little bit unrealistic in terms of what we are trying to achieve in modern healthcare, like Wolverine or Deadpool, who can heal themselves in seconds. But they're healing everything. So they're healing bone, and they're healing part of the blood transport system within the body. How close do you think, realistically, can we get towards increasing the speed of our body's ability to regenerate? And do you think we can do it? Is it something we should do?
Yeah, I'm not sure you want to increase the speed, but you want to improve the outcome. I think that's the more important. So you want that the outcome is perfect. If it takes a bit longer to get there, I would be perfectly fine with that.
So in your case, a good outcome would be less scarring, more in the case of bone, better fusion between different bones,
Yeah,
bone components.
no pain, quality of life.
Yes,
Yes.
lack of chronic issues.
Yes.
Similarly for you, Anthal, or do you think differently about this?
Well, I think we are miles off from any of this like
really
multi-tissue regeneration.
Both of you have disappointed me a lot with those comments. I'll just continue to dream.
I mean, there are also systemic therapies being developed that are not necessarily built like traditional tissue engineering therapies, like where you replace one bit of your tissue, and that might be a good strategy for this. Yeah, of course, I'm biased because I study immune cells, so I think the answer isn't there if we would ever want to get to that. But I agree with Sandra that the most important thing is to improve the quality of life for a patient and not to be like a spare parts factory.
Yes, yeah, I also tend to agree with that.
I mean, in the animal world, there are animals that actually can regenerate tails, whole limbs.
Axolotls,
Yeah,
and
whole limbs.
salamanders,
Yes. So, and I think we can actually, that's not something we're doing, but people are looking at that, like, how can we learn from them,
Yes,
right? So,
absolutely, yeah, because they have the genetic code already
Yeah.
to be able to do that.
Yeah.
Perhaps we have the code in our DNA already, which is dormant. Perhaps. That's something for that's something for number 2 on the Time magazine list, the gene programmers to worry about. In terms of both your fields, I can imagine that do you cross over, do you talk with each other? You're in the same department, the Department of Biomedical Engineering here at TU Eindhoven. How does Anthal's work maybe influence yours and vice versa?
yeah, I can say that so for bone regeneration, inflammation is actually, and in cells from the immune system, so in vivo, that's absolutely necessary. So it won't properly heal without a properly functioning immune system. Nevertheless, in our in vitro system, we're currently neglecting that, apart from the monocytes that we're actually also using to get the osteoclasts, because they also come from that pool of cells. I would say, so the question is a bit, how much complexity do we have to add in order to answer our research questions? But if we really want to fully understand regeneration, I think we need to also integrate the immune system at some point. But then I must also say, where we're at with just 3 different cell types, that's already highly complex. So
Yeah, and it's good to say that, because many people may think that, oh, you're only dealing with 3 types of cells. And one of them, to be honest, has a kind of a contradictory name, because the osteoblasts, if I was been looking at the name, you'd think that, oh, it's a blast, so must break stuff apart, but it's not the one that does that.
No,
It's
it forms,
it
yeah.
forms. So they might be thinking that there's only 3 cell types there, it's not too complex, but it is a very complex system.
yeah, you can start with, if you culture them individually, you just give them all the right nutrients, and they do mostly what you want them to do. But these 3 different cell culture media are different. So if you combine them, which one do you use?
Yes.
Which one do you support?
Yeah.
Do you choose one? Do you mix them? So it adds a lot of complexity already with that. Now we're starting with progenitor cells that first have to become cells. So if we take progenitors for osteoclasts, but use the medium that is favoring osteoblast, they go completely in a different direction.
Yes.
Yeah.
Which can be difficult to control.
Yeah.
Okay.
Yeah.
Anthal, what do you feel that when you think about Sandra's work, how can it influence what you're doing?
Well, quite a lot actually, because I think like these in vitro models are very important to both of us. So we actually have PhD students that are share an office, and they sort of they mingle, and so there we are exploring now what Sandra's group has their work on, for example, the medium conditions that are that you need in the lab and how to control that well. I know very little about that, so in that sense there's immediate crossover. And we also have a link in the sense that our tissues are loaded
Yeah.
So bone is heavily loaded.
Yes.
Cardiovascular tissues are loaded, and that goes for most of the applications we study in our groups. And I think there also is a crossover. So we always use bioreactors, and some questions we ask, we try to answer in the lab are very fundamental. For example, how do certain cells respond to a certain load, like a strain or a shear stress or whatever? And that is basically tissue independent, right? It's fundamental knowledge that can be applicable to different tissues. So I think there also is a nice crossover, yeah.
Okay, so I think actually it's good to actually have an ethical question about this, because of course you're all working with, both of you are working with cells. Of course you get proper ethical approval with regards to this, but then it's more thinking about the effect it can have on society, whether it's ethical and responsible innovation in terms of what you're doing. And it's about making sure that these type of treatments can be available to everybody. So I can imagine that if you're growing heart valves or potentially growing a full replacement bone for whatever reason that may be, for drug testing or for something else, that it can be quite costly and that it can be maybe only accessible to a small group of people. How do we, and you as tissue engineers and maybe the greater tissue engineering community, make sure that your research becomes widely available to everybody or as many people as possible?
Whew, difficult question.
I'm sorry if it's a difficult question.
No, but it's a good one. So first of all, we try to publish open access.
Sure.
I mean, that's one thing, so that people see what we've tried, what has worked, and actually also what has not worked. So if that goes into publications too, and would be more acknowledged, I think that would be helpful for everyone else.
Yes.
To not make the same mistakes again. Well, with the in vitro models, for example, we hope that we can reduce the number of animal tests needed. So that's one thing that addresses your ethical question or ethical aspect as well. So
for society in terms of...
Yes.
Yes.
Yeah. By including human cells, we hope that we're closer to representing the human system. And yes, if you talk about costs and not being available to everyone, yes, that with personalized medicine, that might be an issue. On the other hand, if we can study a few patients, for example, we might get some knowledge out that is actually true for the whole population of patients with that certain disease, for example. So more than just those patients that we have looked at might benefit from it, potentially.
Because it may be applicable to a certain demographic with certain conditions, and there are many people who can be placed in these demographics.
Could be.
Yeah.
I must mention, so we're still away from that, right? That's a bit where I'd like to get towards, but we have not yet shown that we can do that.
But it's good to look ahead,
Yeah,
right?
yeah.
It's always good to look ahead. What about you, Anthal?
Actually, this is a very lively discussion in the field of heart valve transplants at the moment. Because I think it's also because of the cancer therapeutics that are on the market now that are very expensive and not so basically only accessible to wealthy people. What we have done in a project recently together with people in Rotterdam, Erasmus Medical Center, is they developed models to calculate basically how much one of our valves could cost depending on how it would improve the quality of your life. So, and then basically it's simple math. So you know then how much it can cost to first beat existing treatments, so beat an existing prosthesis, but also how to keep it accessible basically.
Sure.
Although I must say, to translate our mechanistic knowledge into a product, you need investors. And in the end, they will pull strings, of course.
Yep.
And what is interesting to say as well for us is that in 2018 there was a declaration signed in Cape Town by a lot of scientists and clinicians that we are, for heart valves, we are mainly focusing on creating aortic valves for patients with calcific aortic valve disease, which is a typical Western world disease because of bad diet, shitty exercise, and you get a bit, you need a new valve at some point. But there's a big, by far the biggest patient population would be in the second and third world countries, so to say, who have a different form of heart valve disease. And that changes the demographic, for example, in terms of age. Those patients have a much wider age range than in the Western world. So to be at least aware of this, it can change your research to, right, not to focus only on the elderly people that need aortic valve transplantation, but also focus on younger patients with rheumatic heart valve disease and replace their valves. We've also been talking about the technology you need to implant a valve, like the hospitals. It cannot be too complex in terms of imaging. We can implant a valve through a catheter, but you need highly complex imaging technologies to position it. Then it will automatically not be available in these type of countries. So these are things that are
it's a difficult question to ask, because you're doing very, very, very interesting and advanced research, and I'm always thinking ahead. Where can we see this in society and have as many people as possible getting access to it? And as you pointed out, it's personalized medicine, because you're taking either cells from a patient to go look at culturing heart valves or culturing a bone or a component of a bone, and that's very personalized.
Maybe some important aspect to add here is also, I mean, what you're looking at, that can cost a patient the life, so to speak. Whereas, you know, a bone fracture in most cases, well, if it doesn't heal, you can still live. So to be very pragmatic, you can still live without an arm or without a leg. It's not a life-threatening thing. It's a bit different if we talk about heart valves or heart muscle.
Absolutely. Just very quickly, for people who are at home, you can use the question balloon in the bottom right of your screen to ask a live question. I have a question just looking off into the future. It's 100 years in the future. Where do you hope that your respective fields will be in 100 years in terms of what you're working on with bone and what you're working on with heart valves? Sandra.
Yeah, good one. Yeah, well, I hope that we understand how bone works. I actually started with teaching lectures on bone and bone cells, and I went to textbooks, and it reads as if everything was known and facts. And the more and more I read, as you pointed out before, the more I see we don't know everything. We know bits and pieces, but we don't know the whole picture. And I hope that if we manage to get there, that we understand what makes something work in a certain way. And that doesn't work with like in vitro experiments alone. We need computational models that are actually able to simulate, to describe what happens, that we in that way can avoid or treat diseases, bone diseases, and to help the regeneration of bone fractures, to support those. I mean, it's not that we can avoid them happening.
Yes, of course, they're going to happen.
But that we can, take away pain of patients and help them have a better life.
Yes.
Yeah.
Anthal? Heart valves in 100 years?
100 years is a long time, so I think we can get something done.
Yeah.
I really hope we have a valve that would be suitable for implanting in children with congenital defects, and that would grow with the child. And I think this should certainly be possible. And of course, because of my love for the immune system, I hope we have unraveled it so far that, and not only heart valves, but that we can sort of harness it to induce regeneration where we need it, also for nerves or cartilage or whatever.
So use it, have it for on our side as much as possible.
Yes, exactly.
And the one thing that I'm hearing from both of you in this discussion is that tissue engineering is a multidisciplinary area, and you need people from all different aspects. I'm thinking material scientists, mechanical engineers, pharmacists, anybody else, biologists?
Yeah, and basically those that can translate the languages between the different disciplines. Sometimes solutions are right next to us in another department, but we're not aware of it because they label it completely different. So we need people who can translate these languages. So, and sometimes we can do things for other disciplines, sometimes they can do things for us, but we don't understand their technology well enough to express what we need from them, or they are not equally good at explaining what they actually can do. And I think this I still see as a challenge. We all talk about interdisciplinarity, but I consider it still difficult, but very nice to see what others can actually do.
Yes.
Yeah.
I think it's also the question, like, it's good to have a specialty, I think. Like, that's something that you can fall back on while being interdisciplinary. So I'm an engineer, you're a pharmacist. So you have to have a specialty, I think. But maybe we are also in the time that tissue engineering can be your specialty rather than a collection of different disciplines only. So I think that is also a very important consideration you have to make when you're pursuing your career in science.
When you're thinking about making decisions. And I have to say that 20 years ago, when I was studying physics, I wasn't thinking about tissue engineering, but if I had the time back around and tissue engineering was a career choice or path I could take, it potentially would have been something that I would have followed.
You still can.
I still can. Yes, that's true. Yes, yeah. I think I'll do a PhD. You can vie for my...
We can combine a project.
There we go. Yes, exactly. I just want to say thank you very much, Sandra and Anthal, for your time
Thank you, Barry.
You're welcome.
talk about your very interesting research here at TU Eindhoven. Just want to also say thank you to Lieke van Gorp of Biomedical Engineering for helping with putting all this together, and also Steffi Hermans of MDR Gravity Program for all the great help in putting together this webinar, How to Become a Tissue Engineer. And it's over to you, because you've heard now from two real-life tissue engineers, and it's over to you to start thinking about the trajectories and the paths that you can take to perhaps become a tissue engineer in the future. I've been Dr. Barry Fitzgerald of TU Eindhoven, and we say thank you very much for tuning in.